Hello, ladies and gentlemen. We are going over your endocrine histology today. This video, bear in mind, we're going to be looking at microscope slides. The microscope slides you can see on your midterms. Bear in mind, they are, if you are face-to-face, no two people's samples are alike, so if you are looking at actual microscope slides, they are legitimately thinly itty-bitty sliced pieces of somebody else's thyroid gland or pituitary gland or whatever. No two people are alike, so the images will not be identical to real slides that you are seeing in the classroom if you are my face-to-face class. If you are in my online class, there is no guarantee about whether or not the images you're going to be seeing for your histology questions are either pictures from the internet and from your other resources like your textbook, your lab manual, or other images that are shown in other PowerPoints for our course, or if you are like the Bing image searches that are linked to you within your course. Everybody's glands are going to look slightly different because every person looks slightly different on a cellular level. This PowerPoint is to give you a rough idea for what to look for when you are taking your quizzes and when you're looking at things, so you know what to look for and don't panic, essentially. So we're going down your lab list and we're going to just go over the big what to look for to identify information kind of concepts. So the first gland on y'all's lab list that you need to know is your pituitary gland. And looking at this, I'm sure you are all daunted by the wall of pink that is in front of you. Your pituitary gland is going to look kind of pinkish under a microscope, but I'm sure you can notice the weirdness that is the fact you have this lovely partition running straight down the middle here, and then this side happens to be a light pink that seems like there's more space between your dots, which are the nuclei of the cells you're looking at, and this side, which is a darker pink color and looks like it's more densely packed with cells. Remember that your pituitary gland has two lobes to it that you need to be able to identify, the anterior lobe and the posterior lobe, so you have an anterior pituitary and a posterior pituitary. Well, if we label stuff, and yes, this label is in French, you don't need to worry about the partition down the middle, that's not on y'all's lab list. But if we're looking at this labeled, because your posterior pituitary has tissue samples that are much more like your hypothalamus, as opposed to your anterior pituitary, which actually synthesizes more hormones and distributes them, the cells are going to look different. Because of this, when we have sliced your pituitary and put it on a microscope slide and dyed it so we can easily view, your posterior pituitary here, which, if you remember, your fancy term for that is your neurohypophysis, because hypophysis is pituitary, neuro is the posterior part because it is closer to the hypothalamus, and the tissue is more neural. Your posterior pituitary here ends up looking light pink and more spacious, as opposed to the anterior pituitary, which has more densely compacted cells, and ends up being a hotter pink color, or a deeper, richer pink. Your anterior pituitary is also called your adenohypophysis because it makes more hormones that will affect things like your adrenal glands, and hypophysis, pituitary. So if you ever look at a microscope slide, right, and you can automatically immediately tell there's a weird partition somewhere in it. Maybe they zoomed in so it's not even, but it looks kind of like this square that I'm kind of outlining. And where you see a weird partition and light pink on one side and darker pink on the other, automatically you should know that's the pituitary, because your pituitary is split in half between light pink and dark pink. And forever and always, your pituitary is split in half between light pink and dark pink. It's the only gland that's going to have this weird partition and be half and half color, right? So that's your first easy step, and light pink is your posterior pituitary, dark pink is your anterior pituitary, right? Zooming in even closer, right, we can see the darker pink versus the lighter pink here, and this is zoomed in mostly on your anterior pituitary. I'm not going to talk much about the basophils now because you guys are going to get a separate video about blood cells, and basophils are a type of white blood cell that you need to be able to label. When you guys see basophils, they are far more likely to be in like a blood scraping or a microscope slide of blood, so you can see the different blood cells. So we're going to skip over your basophils right now. You're more than likely, like, I am 95% sure you are not going to be asked to identify a basophil from the pituitary gland. But this brings us to our two other types of cells in your anterior pituitary, chromophobes and acidophils, right? In Latin, if you see something that ends with phile or phil, it means you have an affinity for that, or you enjoy it a great deal, you love it. So a bibliophile is the word that describes someone who loves books. These cells are called acidophils, meaning these cells have a strong affiliation or affinity for acids. Because of that, they end up with a darker color when dyed. They end up being this richer, darker pink that give the anterior pituitary its color. On the flip side of things, the opposite of a phile or a phil for something is a phobe. A phobe or phobia means you have an aversion to it or a fear of it. So, for example, if you are an arachnophobic person or you have arachnophobia, you are scared or have a fear of spiders, right? You guys have heard these words before. So phobe means aversion to or fear of. Chromo or chrome is the Latin word for color. So if you've ever wondered why Google Chrome has that rainbow logo, it's because they named it after the word colors for chrome, right? So a chromophobe is a cell that has an aversion to colors. And as you can tell, this chromophobe here is almost clear in color. You can see the nucleus, but the rest of the cell doesn't really have much color, and it looks like the rest of the world around it. So the two types of cells within your pituitary that you need to remember, the darker colored ones are acidophils, the clear-looking colorless ones are chromophobes. Okay. When we zoom in on your posterior pituitary gland, nobody was creative when they thought of this name. Each of these individual little cells here, you can see the nucleus and then you can see the rest of the cell outlining them. Right? Site is your Latin term for cell, and then pitua is short for your pituitary gland. So literally, the cells you're looking at here from your posterior pituitary can all be summarized as pituocytes, which literally translates to cells of the pituitary. We're moving on to your next picture, or your next gland, right? Your next gland is also a pretty easy gimme one as far as microscope slides go. If you didn't know what this was and you couldn't see that I have labeled this as your thyroid, and you're just looking at this weird mess of a pink picture, it looks like dish soap, right? It looks like the soap that you do when you're doing the dishes. You have all these big bubbles and a bunch of little cells in between, but you have a bunch of bubbles, right? This is your thyroid gland. Your thyroid gland is always going to look like soapy water. It's probably going to be pink, so it'll be pink soapy water. But your thyroid gland always looks like soapy water. If you are looking at a microscope slide and you see soapy water, immediately you should think, oh, thyroid. Right? So there's a few things you got to know about these the thyroid. You have 4 things on your lab list that you need to know. Right? First of all, you have the bubbles here. The bubbles are called follicles. A follicle is a tiny little bubble or balloon inside you that's going to store some chemical or store information. We'll get into that in just a second. But these bubbles are all called follicles. Right? So we know that the bubbles are called follicles. The other 2 types of cells that you have to worry about are the cells that line the bubble or that are around the follicle, and the cells like these ones where my mouse is right now that don't touch a follicle. These cells do not touch one of the bubbles. So you have cells that touch the bubble and cells that do not touch the bubble, right? So they have been called follicular cells if they touch the follicle. So if these cells here are lining a follicle or touching the bubble, they are called follicular cells. That's what this big black arrow is pointing to. If the cells are not touching a follicle, meaning they're like in this area right here where it's clearly not touching a bubble, over here again, clearly not touching a bubble, that kind of thing, they are called parafollicular cells. Para means near. So essentially you have your Latin terms meaning follicular cells for the cells around the follicle, and the near follicle cells for the cells that are near the follicle cells. So you have follicular cells around the follicle and parafollicular cells elsewhere. So all of these cells here are parafollicular cells, and then you have follicular cells around this follicle. The 4th term that you guys have on your lab list can be one of 2 things. I am going to edit your lab list for this. And here we have another picture where you have the follicular cells labeled and the parafollicular cells labeled. These follicles fill with a chemical that will become thyroid hormone inevitably. We'll discuss this a little bit more in lecture. But the chemical that is inside these follicles will inevitably, with iodine and some other means, become thyroid hormone, and this is where your body stores it. That chemical or protein complex is called thyroglobulin. Thyroglobulin is the word that is on your lab list. Right? However, if you are trying, and if you read this in your lab manual also refers to this as thyroglobulin. However, if you are reading in your lecture book for this, and you're reading in the OpenStax chapter, and the PowerPoint for your OpenStax lecture class, instead of referring to it by protein name, the textbook and lecture class refers to this as the collective term for this type of proteins, which is colloid. In my class, I don't want you guys to be confused between the 2. I will accept both answers for this for my class, as far as I'm concerned, because it's not your fault that the textbook company we use for lecture refers to these proteins by their collective term as colloid, and that our lab manual is very specific with which protein it is, which is thyroglobulin. I don't care which term you choose to memorize, I accept them both. The follicles here are going to be filled with colloid or thyroglobulin. You guys can use whichever of those 2 terms you want from me. Right? So follicles are the bubbles. They are filled with a liquid protein called colloid or thyroglobulin. And then the cells that surround the follicles are follicular cells. And the cells that do not touch the follicles, right here, are parafollicular cells. The next thing on y'all's list are the tiny itty-bitty glands on the tips of the wings of your thyroid, called your parathyroid glands. You can look at this slide and you probably immediately wouldn't know what you're looking at, but you see dish bubbles right? If you see dish bubbles, you know that you are looking at thyroid. And if you look closer at this slide, you can see that there is this happy little differently colored pink cluster in the midst of the rest of these cells out here. So you have more purpley cells way out here, but you have a more pink cluster right here in the middle. This pink cluster right here in the middle is your parathyroid gland. You know you're looking at your parathyroid because you know you're in the vicinity of your thyroid because of those dish bubbles. So you are identifying parathyroid at first glance by its proximity to what you can easily identify as the thyroid. Once again, thyroid gland, you have the dish bubbles and soap all throughout here, but this corner right here is the parathyroid gland. So it's distinctly cut from the thyroid. There's some space here for connective tissue, and you have the parathyroid gland immediately here. So you can identify this part as thyroid pretty straightforward and easily. Parathyroid, here we go. Right. Once again, this is an even greater close-up. You can see bubbles from the thyroid, and then you have this vast area here that does not have bubbles in it, which would be part of the parathyroid. The two cells you need to recognize from your parathyroid are the chief cells and oxyphil cells. These larger pink cells here that look like they have more bulk in the cell because the little black dots are the nucleus, and you can see the big cell around the nucleus. So these look like bigger cells, and they are the ones that are more pink in color. These ones are your oxyphil cells. The other cells of the parathyroid that are not oxyphil cells are your chief cells. Chief cells can also be called principal cells. Both of those are on your lab list. I will accept either of those two answers. They are both on your lab list, whichever one you put down if you are asked these questions. And so here we have an unlabeled one, right? Which you can see. Chief cells are going to be the pinker ones. Oxyphil cells are going to be the pinker ones. Chief cells are going to be the others. Right, the next gland you guys have down on your list is called the thymus. The thymus is pretty interesting as far as cells in histology go. And your thymus, when looking at it under microscope slides, can be either pink-purple or it could be a reddish color. It just depends upon the dye that was used. So don't let the color trip you up on this one. What's more important for this is you have to know the parts. So your thymus here is divided into lobes, just like streets and buildings for a dormitory, because remember, your white blood cells are going to be growing up big and strong in your thymus so they can do your immune system's job. So this right here, like you can see this septum here. So you have this chunk or lobe here. You can see another lobe here. But these are under a microscope. They are tiny itty-bitty lobes, so they are called lobules. So all of these little separate buildings or areas that you would see are lobules. Right? So this is a lobule, this is a lobule, 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 lobule. There is connective tissue holding all of these lobules together. And that connective tissue we call the septa, which is the word that is on your lab list. Septum is the singular. So there's multiple of these, so you say the word septa. Septum is the singular. If you need a more direct example of what a septum is, if you're looking at your nose, that little bridge of skin that separates your left and right nostril, that sometimes people get pierced, is called the nasal septum. So that is the nasal septum. It's called a septum piercing if you get it pierced. It's a septum. It separates the left and right nostrils, just like these septa separate lobules. Another thing to note about this is you can see that there is a lighter, stringier kind of middle and a denser, more meaty looking outer part of all of these lobules. That is going to be a theme of anatomy and physiology here. You're always going to have, or not always, but a number of different organs have lighter, stringier centers, and then more meaty, dense outer parts. Your kidneys are a good example of this. Your adrenal gland also has this. We have terms that describe the meatier outer bits and the lighter or stringier inside middle. The lighter middle is always going to be called the medulla. Whenever we have to deal with this, be it kidneys, lymph nodes, adrenal glands, or this microscope slide of your thymus when we're describing the lobules, the middle, lighter part is always going to be the medulla. The meatier, denser outer part is always called the cortex. So know the words cortex and medulla. Those questions on y'all's lab quizzes and your final exams and midterms are going to be the gimme questions that you're kind of expected to just get right for free points, if that makes sense. So cortex is the outer meaty bit of all of these lobules. Cortex. Medulla is the inner lighter area. That brings us to three things. Well, first of all, these are red dyed slab, or this is your labeled slide, and these are red dyed ones. So you can see that each of these is a lobule. You can see the inner lighter area, which is the medulla of all of these lobules. Right, medulla, medulla, medulla here. You can see the connective tissue of the septa, or the septum. And you can see the cortex, which is the outer meatier bit. Right, so cortex is outer meatier bit. There is also usually a protective covering around these organs called the capsule. But this is your labeled slide for this. Once again, you have a labeled slide here where you have the septa labeled, and you see the medullas and the meatier cortexes within the confines of all of these different lobules. This one is a young person's thymus. Your thymus is actually larger when you are a tiny child as opposed to when you're adult proportionally to the rest of you. And that's one of those biological imperative things, right? Children put everything in their mouths. It's what they do. When you're a toddler, you wander around, crawling around, and you just like reach for things and put it in your mouth. And because your thymus has to do with your immune system, you need a more amped up immune system when you are in the phase of your life where everything goes in your mouth. Adults, though, we're used to not putting everything in our mouths, so we don't need as big of a thymus anymore. So once again, labeled. That brings us to another word on our list, right? Or another couple of words on the list. The regular cells that you can see are called reticular cells. So all of these regular cells that you can see with the nuclei and the other parts are all called reticular cells. Lymphocyte, which is also on your lab list, is another specific type of white blood cell. You're going to be asked to identify those as a white blood cell on like a blood smear picture. Don't worry about those ones for right now. Because remember, lymphocytes use your thymus as a dormitory. They grew up here. But you have these little clusters that look like hurricanes throughout these pictures. Again, you'll see some of these like right here. You can see some of these right here on this one, like these little hurricane dots in the middle of the medulla. You see more of them in that first picture I showed you. You have two potential names for those little hurricane-looking dots, right? Because you have two names for them on your lab list. They can either be called thymic corpuscles, Or you could try to remember their name by the angry German name, which is Hanselkorpuskel. I prefer angry German because it brings a hint of, like, fun or amusement for me, right? And Hansel's corpuscle is just so much more fun to say than thymic corpuscle. So these are called your Hansel's corpuscles. They are just little clusters of cells that look like hurricanes in the middle of the medullas. They are your thymic corpuscles or your Hansel's corpuscles, whichever you would like to call them. You can see more lymphocytes on this super close-up of the cortex of your thymus. Reticular cells are the ones that look kind of squared off. The lymphocytes are the ones that look like cells that look like a giant eyeball. And that brings us to your adrenal gland. Your adrenal gland is a colossal pain in the rear end. Like, I'm sorry, I cannot sugarcoat that. Your adrenal gland will be a pain in the rear. You guys have to know the layers of your adrenal gland because your adrenal gland comes in layers and it is painful. So here we can see the outer capsule plus the cortex layers. Right. Oop. Here's a zoomed out version. And this is, yes, of a horse adrenal gland. Just depends on what slide on which campus you are looking at. But your adrenal gland has the stringier middle medulla area, and it has a large cortex. That cortex is divided up into other layers, which you guys will need to know. And remember, each layer gives off a different hormone. Right? So you have the protective capsule layer of your adrenal gland. And you have this middle medulla area that looks like it's stringier with more space between the cells. From the medulla comes epinephrine and norepinephrine. That's your fight or flight response. So you have epinephrine and norepinephrine coming from the medulla. And then you have this cortex here. The cortex here has different layers. You have the zona glomerulosa here, which is where mineral corticoid hormones such as aldosterone come from. So mineral corticoids like aldosterone that help control your blood pressure come from the zona glomerulosa. The next layer down is the largest layer of the cortex, called the zona fasciculata, or the zona fasciculate, depending upon which textbook you're looking in. And that gives off glucocorticoids like cortisol. Cortisol is the main hormone you would need to know from that region. Cortisol you can buy over the counter. It's that same stuff that when you get into poison ivy after doing yard work, you buy it over the counter and then rub it over your poison ivy because it makes the poison ivy site unswell and stop itching. Your body makes some degree of cortisol to deal with inflammation and itching and pain, and this is where it comes from. It comes from the zona fasciculata in your adrenal gland. And then you have the area of more spread out but more rectangular cells that are near the medulla, called the zona reticularis. And the zona reticularis makes gonadocorticoids. Gonadocorticoids are hormones. Gonadocorticoids are hormones like androgen or estrogens. Androgen is a hormone, or is a hormone base that your body makes that is the sexless sex hormone. Androgen does the job of whatever needs to be done, and it can easily chemically turn into either estrogens or testosterone. So ladies, because we do not have testes, and we don't make testosterone from testes that we don't have, the testosterone that we have in our bodies, that we use to help build muscle that give us sex drive, generally comes from the androgens that are created from this zona reticularis. Because the androgen, turns into or chemically one-ups itself into testosterones. On the flip side of things, gentlemen do not have ovaries to make their own estrogen. So their androgens, when they need estrogen to help with fat, like fat-soluble hormones and skincare and other such things that's in their bodies that estrogen is used for to balance out, their zona reticularis and androgens will make the estrogen that their body needs. But an androgen is a hormone that will help and can turn into whichever side of those hormones your body needs. And that's what the zona reticularis makes. It creates androgens. So once again, you have 5 layers of your adrenal gland. You have the capsule on the outside that's protecting it, the zona glomerulosa, which makes aldosterone, zona fasciculata or fasciculate, that makes cortisol, the zona reticularis, that makes androgens, and then the middle medulla of your adrenal gland that gives you your adrenaline rushes by making epinephrine and norepinephrine. Right? If we are looking at these underneath a microscope, chromaffin cells are the cells that are going to have deeper coloration inside your inside your adrenal glands. They are in the medulla area. Otherwise, you're good on your adrenal gland now. That brings us to your pancreas. We only have 3 more glands to go over in this video. Huzzah, guys. So remember, your pancreas is one of those organs that you want to keep happy. Make sure they are pleased and get paid well and taken care of, because your pancreas pulls double duty for you and works full-time for 2 different systems of your body. Right? So your pancreas works full-time for your digestive system, and it works full-time for your endocrine system. So when looking at it under a microscope, it's no surprise that you can see bursts in different areas for each job, right? Your pancreas has to be well organized that way. So if we're looking at your pancreas and it's been cut long ways, you can see this ductwork. Duct, D-U-C-T. So you see this ductwork, and that ductwork is usually attached to the digestive system jobs of your pancreas, because remember, your endocrine system is ductless. So hormones are just dumped into bloodstream. That's going to get carried away and dealt with body-wide. But your digestive system is not body-wide. It is not systemic. It is an enclosed system of itself. So the areas around the ductwork are going to generally be more digestive system oriented. Right? So we have an interlobular duct and an intralobular duct. You don't have to be able to identify the difference between those two, but you have ductwork here. You see some blood vessels because, of course, there's blood vessels in there. Most of the cells that you are seeing that are not in these bursts of light pink circular areas, these clusters here, are called acini cells. Acini or acini cells, however you would prefer to pronounce them, are the ones that make bile for your digestive system. So the acini cells in your pancreas are the ones that do the digestive system jobs. On the flip side of that, you can see these circles, or these lighter pink nodes, for lack of a better word, within your pancreas. You have 2 potential names of those little nodes. They are either called your pancreatic islets, or they are called your islet of Langerhans. Once again, with the angry German, makes it easier to remember and more fun to say. Your islet of Langerhans here are the parts of your pancreas that work with your endocrine system. So they are the parts that make your hormones that we're worried about. Right? So islet of Langerhans is right here. They're the part that make insulin and glucagon. And then the pancreatic acini cells are the ones over here that make bile for your digestive system. I know your lab list also tells you need to know beta cells and alpha cells here. There is absolutely no way you can tell the difference looking at these to know which one is a beta cell and which one is an alpha cell. Because alpha cell, they're defined by what they make. And you can't see the chemicals that are being made by those cells in a slide. Alpha cells make glucagon, beta cells make insulin. That's the difference between them. That's more of a lecture-based question. But no, I would know that your islet of Langerhans here is made of alpha and beta cells. You're not going to, like you in my class, you're not going to be asked to identify the difference between an alpha and a beta cell. And here we have testes. This may come as a shock to some of you, especially my gentlemen, but the testes are actually a cluster of tubes. So if you see this weird conglomerate here of what looks like a lot of tubes and a lot of space that's just holding these tubes together, congratulations! This is what your testicle looks like under a microscope. You can also easily tell that this is a testy because the way you make sperm in your testes is you have these tubes, and we'll get into the names for everything when we get to the reproductive system. Don't worry, but right now it's an overview. But you have these tubes, and from the walls of these tubes, right, you grow the cells that grow into sperm. When these cells are adult enough, they will, and like ready, almost ready to be freed, to be stored by your body, they will develop tails and then release from the walls of these tubes. It's how sperm are created and made. So if you're looking at this and it looks like you're observing a series of tubes and some of these inner cells seem to have stringy tails here, congratulations, you're looking at sperm. This is a testy, right? Testes make testosterone, and you are looking at tubes creating sperm. And for your last gland that you needed to know for this system, right, is the ovary. Ovaries are similarly easy to be able to name. Most of your ovary slides you're going to see are going to be very specific, and you're going to be able to see a decent bit because it is part of your reproductive system that you need to be able to label the differences in the development of these eggs. So the last unit we'll get into the difference between like a secondary ovarian follicle versus a primary follicle right here and all that kind of jazz. You don't have to worry about that for your endocrine system right now. The most important thing for you guys to know and understand for this unit with endocrine system is ovaries make estrogen and progesterone. And what you're looking for is you see all this tissue-y bit stuff, and you can see eggs here. So these are just clustered eggs. And there's usually one egg that has been chosen and is the growing one. And it's going to have this large bubble area around it that's acting like an actual egg. So it has a buffer to it, so you can see this one larger developing egg right here. And the oocyte in the middle. Right, with all of these other smaller eggs that have not developed yet off to the side waiting for their turn. So once again, over on this side, you have oocytes right around here. So eggs, or oocyte is your fancy word for egg. And then you have the chosen one, and you have the oocyte here with its space around it and the body building its nice little protective cluster and wall around it for protection. You will easily be able to see eggs and one super deeply developed egg on your slides of an ovary, if you are shown an ovarian slide. And that should be all, my lovelies. Right? I hope this helped clear up some stuff for you guys as far as your lab list and lab stuff goes and what to look for histology. I know histology can be intimidating. Just Google search some of the terms on the histology, like ovary slides, ovary histology, testy slides, testy histology. and see what you can label. See what Google or Bing brings up for you. I know you have a direct image search to Bing in your lab section, but I prefer Google image search. Whatever you guys do, like, just quiz yourselves, and I hope this gave you a decently good overview for what to look for. Have a wonderful afternoon or day. I'll see you guys next time.